Hi and welcome to my poster. I'm Matthias Lienhard and here I present IsoTools, my Python package for long-read transcriptome sequencing analysis. IsoTools is designed as a Python API to explore and analyze and visualize long-read transcriptome data, such as from PacBio IsoSeq or Oxford Nanopore. It starts by importing aligned long reads into an efficient tree-based data structure. In this step, the transcriptome is reconstructed, quality control metrics are computed, and novel transcripts are classified according to the relation to known genes and transcripts. The user can then access and explore the data, use statistical methods to extract relevant information, and then export or visualize the data. For experimental planning, I implemented a statistical model for the required depth of sequencing. Assuming you're interested in a transcript with an estimated abundance of, say, 1 transcript per million, and you want to observe at least, for example, 4 reads for this transcript with at least 90% probability, this model can compute the required number of reads. To identify and handle technical artifacts, I implemented several quality control measures. And these can be used for the flexible query syntax to define exactly what transcripts to use or to export. The transcripts that are not in the reference are classified into 19 categories, how they relate to known genes. Here in this example, the most frequent one is novel combination of known splice sites, but there are also novel exons, for example, or fusion genes. The transcript structure of a gene is encoded internally in a graph that is used in several methods for efficiency. The most remarkable use is the definition of alternative splicing events. By searching for specific substructures in the graph, alternative splicing events can be found and then classified into exon skipping, 5' alternative splicing, 3' alternative splicing, mutually exclusive, and intron retention events. And then they can be quantified with a % spliced index. There's also a statistical test for differential splicing between groups of samples. Last, IsoTools implements a bunch of visualization methods for transcriptome-wide statistics, as here the transcript length distribution, quality control metrics, but also sashimi plots, for example, and 2D embeddings of alternative splicing events, as for example down here. For IsoTools, there is a preprint on bioRxiv describing these and more methods in great detail. IsoTools is open source on GitHub, but is also on the Python package index, PyPy, so it can be easily installed with the pip command. There's also extensive documentation of the API, including tutorials with case studies on Read the Docs. If you have any questions, don't hesitate to contact me. And thank you for visiting my poster.